today's episode of Myeloma Crowd Radio, a show that connects patients with myeloma researchers. I'm your host, Jenny Ahlstrom, and I'm joined today by my myeloma friends, including Pat Killingsworth, Gary Peterson, Cynthia Chemlowski, and Jack Aiello as co-hosts. This is the fourth in a very important series featuring the Myeloma Crowd Research Initiative. For the first time, patients, including you, are teaming up with myeloma researchers to find and fund the ideas in myeloma that could have the greatest impact for the next generation of myeloma therapies. We decided to go after high-risk myeloma. For patients that have no viable options today, either because they have high-risk features or are relapsing or refractory to existing drugs, we need new options. And if we can find a solution for high-risk patients, it's highly likely that it will work in medium or low-risk patients. It's also common to pick up more aggressive genetic features as the disease progresses. So, unfortunately, many of us become high-risk after our myeloma has relapsed multiple times or current medications become ineffective. We're now in stage 3 of the Myeloma Crowd Research Initiative, or we call it the MCRI. In stage 1, we asked researchers around the world to submit their proposals, and we received back 36 high-quality letters of intent. In stage 2, we had those proposals scored by our scientific advisory board. They selected 10 proposals, which were they believed were the top proposals, and we are now holding Myeloma Crowd Radio shows so that you can become involved. We want you to understand the proposals, so please listen in, ask questions, read the transcript after the show is posted, and share with your friends and family. After the full proposals are submitted, the Scientific Advisory Board and Myeloma Patient Advisory Board will together decide on the limited number to fund through patient-driven campaigns, and we will need your help to get the word out and share the very amazing work that's being done. Now today, if you'd like to donate, we have a donate button on the Myeloma Crowd site, and you can help us start to donate for this very unique endeavor. The specific projects will come later, and we will allow time for these full proposals and all the shows to be completed. Now, we are very privileged today to have with us Dr. Bruno Paiva of the University of Navarra in Spain. And because Dr. Paiva is joining us from Spain, we may have a little delay until he joins us. So I'm going to give us a little introduction for Dr. Paiva. Dr. Paiva is Scientific Coordinator of the Flow Cytometry Department in the CIMA Lab Diagnostics Group at the University of Navarra in Spain. He teaches courses regularly on cellular biology, techniques of quantification and analysis, new treatments in hematological cancers, inflammation as it relates to the blood, and minimal residual disease. The Spanish group with which he has worked has performed very extensive work, appreciated by the entire myeloma community and myeloma practitioners on the flow cytometry test as it relates to minimal residual disease. He specializes in understanding the heterogeneity or biological complexity of MGUS, smoldering myeloma, active myeloma, and how it evolves. He's involved in both the detection and classification of myeloma and has written numerous papers on the topic, writing or speaking on the subject over 140 times. He's also been principal investigator on clinical trials utilizing immunotherapies, including the SAR anti-CD38 monoclonal antibody. He participates as an ad hoc reviewer for Blood Cancer Journal, Blood, Pathobiology, Leukemia, and several other publications. He's received premier ASH Abstract Awards in 2014, 2013, 2012, and a Young Investigator Award by the IMF, to just name a few. Dr. Paiva, welcome to the show.
Thank you so much. I'm sorry for the slight delay. I had some technical difficulties, but I think now it's all set.
Well, perfect. We are very thrilled to have you. So on the call live, we have Cynthia. We have Jack. Let me bring on Gary. Okay, Gary, you should be on.
Excellent. Good afternoon to all.
Yes, I am.
Okay. Well, for those who are joining live, we as co-hosts, we'd ask you to keep your microphone on mute unless you're ready to ask your question. So, Dr. Paiva, maybe you can give us first some background about the study of minimal residual disease, why it's important in myeloma, what you've learned so far, and I know this is background because we definitely want to get to your research and what you're studying now, but if you could give us a little background, I think it'd be very helpful for patients.
Absolutely. Plus, it's really an emerging field, which is quite natural, and in fact, the implementation of new biomarkers takes several years. And if we look, for example, to other diseases such as acute leukemia, the concept of MRD, which is now fully established, took a long time. In multiple myeloma, unfortunately, until 5-10 years ago, there weren't many therapeutic options. The options available were not tremendously effective, and therefore complete remissions were rather scanty. Only a few patients achieved deep levels of remission, and consequently also survival was limited at the time. Nowadays, and as we are probably all aware, there are many more and very effective options to treat multiple myeloma. These produce nowadays much higher rates of complete response, which for both younger transplant-eligible patients as well as for elderly non-transplant candidates are now in the range of 50%. Therefore, you can see the dramatic evolution and a fortunate evolution on the treatment of the disease. We can also see a dramatic evolution in our understanding of the biology of the myeloma plasma cell, also the biology of normal cells, for example, mesenchymal stem cells. We also see a great improvement in patient differential diagnosis. We now have better tools to differentiate between treatment-requiring myeloma, ultra-high-risk myeloma, smoldering myeloma, high-risk smoldering myeloma, MDAS. Also better tools to predict the risk of newly diagnosed myeloma patients, discriminating between those with standard versus high-risk disease. But in contrast, and for many years now, there has been no evolution in how we measure response to therapy. While before there was really no rationale to improve the sensitivity of the techniques used to monitor response to therapy, nowadays, given the efficacy of the novel agents and more complex treatment schemas, it becomes mandatory to measure a response to therapy using very sensitive tools in order to understand and to identify those patients that have truly responded to chemotherapy and achieved deep levels of remission from those patients which are more chemo resistant. And the identification of both will be crucial as we move along into precision medicine, and we'll try to avoid both under as well as over treatment. So here relies the importance on minimal residual disease.
Yeah, when I know the International Myeloma Foundation has the Black Swan Research Initiative, and we're supportive of the minimal residual disease testing and are supportive of that. And I didn't fully understand the importance of minimal residual disease testing until I talked to Dr. Landgren, and he was saying how it will tailor treatment and allow more of the precision medicine to say, okay, if you haven't responded, this is why, and then you can customize treatment for different individuals.
Absolutely. That's the final goal and purpose of all the years of intensive research. Many groups, I would say in particular, but not the only one, the Spanish myeloma group, is really devoted to the field of MRD.
Oh, no.
technological development. Then, after developing the technology that at the time was pretty much low sensitive, it emerged as one of the most important prognostic factors. In our experience, the most relevant prognostic factor. And now we are in the second half of this match, in which after the initial development of the techniques to monitor MRD, we are now making a final fine-tuning to really optimize the sensitivity of the methods, also to standardize the methods for a global and wide application of MRD technology, ultra-sensitive MRD technology, and this not anymore as a plain or a simple prognostic marker, but also as a diagnostic tool to really try to optimize the management of myeloma patients. It should be emphasized that, again, precision medicine or treatment tailoring can never be decided on a single biomarker, rather in a combination or a constellation of biomarkers that provide complementary information. But we certainly believe that now, with more sensitive and standardized MRD techniques, MRD can become indeed that biomarker to help optimizing the management, the clinical management of myeloma patients.
Okay, perfect. Can you explain what the MRD test originally was looking at, and then how it's evolved, what it's looking at now, and kind of explain that evolution of that test?
Absolutely. Originally, MRD testing was performed mostly with two different techniques. Multi-parameter flow cytometry that looks into live plasma cells and discriminates between normal plasma cells, which all of us have in our bone marrow as well as in secondary lymphoid tissues, peripheral blood, versus myeloma plasma cells, which have a different phenotype. So the way we discriminate between normal versus myeloma cells is according to the presence of aberrant phenotypes, aberrant antigen expression in the surface as well as inside the myeloma plasma cell. Alternatively, there's also molecular PCR that is capable of detecting within the DNA of patients' bone marrow samples, is capable of detecting the clonotypic sequence of the monoclonal immunoglobulin that characterizes all myeloma plasma cells from every single patient. It's like the passport of the myeloma plasma cell. Now, both techniques have been there for myeloma as well as for other hematological malignancies for now more than 10, perhaps 15 years. PCR, in particular, the more specific PCR approach, which is called ASO-PCR, a real specific oligonucleotide PCR, has perhaps been more sensitive than multi-parameter flow, but as the handicap, the drawback of the applicability, because myeloma is an end-stage B-cell disorder, is also characterized by a higher rate of somatic hypermutations. And this clearly limits the applicability of ASO-PCR to monitor MRD in multiple myeloma. By contrast, multi-parameter flow from 10 or 15 years is much more applicable to virtually all the patients, but was less sensitive. Nevertheless, all the available data, and we now have more and more data and consistent data using both low sensitive flow and ASO-PCR, show that both strategies afford clear prognostic value even when performed among patients in complete response. And in complete response, theoretically, you don't have disease anymore, but by using more sensitive methods, residual disease can still be identified. Now, more recently, there has been a dramatic evolution in both approaches. First, ASO-PCR has moved from a single monitoring of the unique clonotypic immunoglobulin sequence into high-throughput sequencing or next-generation sequencing. This allows to monitor MRD by molecular approaches with the same sensitivity, perhaps even slightly higher as compared to ASO-PCR, but the applicability, since now we measure all the immunoglobulins present in bone marrow samples, the applicability is much higher, in the range of 90% of the patients. Regarding flow cytometry, we have moved from measuring only 4 proteins at the same time into 8 or 10 proteins simultaneously, antigens, and we have moved from measuring 200,000 cells into 5 million up to 10 million cells. This means that flow is now much more specific, much more sensitive. It has moved from a sensitivity of 10 to the minus 4, that is the identification of a tumor cell within 10,000 normal cells, up until 10 to the minus 6, which is the identification of one tumor cell in 1 million normal cells. Plus, with the availability of better cytometers to measure, again, 8 or 10 antigens simultaneously, this means that the technique is applicable to virtually all myeloma patients. So this has been, I would say, one technological revolution. There is a second one that should come in the next 3 to 5 years, which is to implement, in addition to either flow or molecular techniques that measure MRD in the bone marrow, to make synergism of these techniques with other imaging techniques such as PET-CT or whole-body MRI that could measure minimal residual disease in other sites of the bone marrow that could not be evaluated with a single bone marrow aspirate, as well as extramedullary disease. Altogether, this means that even though so far many studies from different cooperative groups in different countries have shown the great clinical value of MRD studies, perhaps we still don't fully understand and realize the clinical significance of MRD in myeloma, since we now have much better tools to monitor sensitive response to treatment.
Okay, so in the evolution, it sounds like you're combining the flow cytometry with the PCR tests, which I understand are the gene expression profiling tests. Is that correct?
Not exactly. Well, during the spectrum of the evolution of disease, many different techniques or tools should be employed according to the specific clinical question. For example, initially karyotyping, then FISH, then gene expression profiling, now deep targeted sequencing or panels of mutations are being employed or have been employed to determine the risk of the patient at baseline when the patient is newly diagnosed. By contrast, these type of techniques have been more used to monitor response. Then again, different techniques are complementary along the spectrum of the disease.
Okay, so it's more of a monitoring tool during the course of treatment.
It's a diagnostic tool. It is also used at the initial screening. For example, even when there is only a suspicion of a monoclonal gammopathy, and you need to establish clonality in the bone marrow or to detect circulating tumor cells in peripheral blood, or to make the differential diagnosis between for example, two different monoclonal gammopathies, as could be myeloma or Waldenstrom's macroglobulinemia. So there is an application for flow at the beginning, as well as particularly during treatment to assess response.
Okay. All right, that makes sense. So your study, okay, go ahead. Cynthia, do you have a question?
Yeah, I have a couple questions. Can I butt in because I do have to return to a meeting?
Sure, go ahead.
Okay, so my understanding, Doc, is that you're developing this device that's going to be much more sensitive in testing MRD than any other device that's already available on the market. Is that correct?
Indeed, the assay, it's not really a device, but it was an optimization of the already available devices. And so far, with 1.5 to 2 years of research between different groups, certainly under the umbrella of projects such as, or research programs such as the Black Swan Research Initiative, now those techniques are much more sensitive. So the sensitivity is already there.
And then this device will then be readily available in labs throughout different countries that many clinicians can use it to test for MRD negativity and positivity. Is that correct?
Indeed, as we believe that MRD monitoring will become more and more frequent in multiple myeloma, given the higher and higher rates of complete response after initial therapy, as well as after salvage therapy for relapsed refractory patients. We believe that MRD will become more and more implemented. And this is, of course, to be used worldwide.
Okay. Now, when I was reading your letter of intent, it seemed like there were different groups of people, and that high-risk multiple myeloma people who, after treatment, still have MRD positive results in their testing are more likely to progress than standard risk multiple myeloma people who still have MRD positivity in the results. Is that correct?
Yes.
So what you're trying to do is look at those minimal residual disease clones that are left over after treatment and compare those clones from the people who have high-risk disease to standard-risk disease to see what makes their clones more aggressive than the other people's clones?
You are correct. We have two, there are different objectives in this letter of intention research program, but bottom line, there are two clear and well-defined objectives.
Okay.
The first focusing on high-risk patients that reach MRD negativity. We want to understand what is the clinical significance for high-risk patients to reach MRD negativity with ultra-sensitive techniques. Does this mean that high-risk patients reaching these levels of remission will have a similar outcome as compared to standard-risk patients also reaching MRD negativity? If this is true, can MRD negativity become a surrogate marker for improved survival in high-risk patients? If this is true, can MRD negativity become a clinical endpoint for high-risk patients? And if this is true, can MRD negativity become a surrogate marker for prospective determination of the efficacy of novel agents before waiting for the long-term overall survival to see differences between two treatment arms in, for example, a randomized clinical trial? So this is a clear objective in high-risk patients reaching MRD negativity. Alternatively, or conversely, there is a second well-defined objective, which in fact arises from our own observations, which is that patients with standard and high-risk disease with the same level of persistent minimal residual disease after therapy. Theoretically, their outcomes should be identical because they have achieved the same level of response, but it is not. And unfortunately, patients with high-risk disease at diagnosis have inferior outcomes even though they have the same number of residual cells after therapy. And this is the main reason why in the next couple of years we want to study the biology of the resistant cells after initial therapy. And it's important to emphasize that, and this is really for the first time ever, we don't want to study only diagnostic cells. We don't want to study cells at the moment of relapse, refractoriness. We want to study those cells, those very few cells that are capable of resisting to upfront therapy. And we want to understand the biological differences of these cells between standard versus high-risk patients in order to understand why in the later outcomes, unfortunately, are still inferior.
Okay. So you're going to be, for the resistant cells of those high-risk myeloma patients that you checked after MRD, you're going to start probing them and looking for some biomarkers that might be on those cells that maybe people who don't have high-risk myeloma, who have leftover disease, don't have those biomarkers.
Yes, and this is how, as Jennifer mentioned at the beginning, this is where really next generation flow and next generation sequencing or immunophenotypic and molecular technologies really merge and have combined value. So one of the advantages of monitoring MRD with flow is that we can not only monitor the disease, but we can also sort with high purity those very few residual cells. After sorting, we can perform molecular assays on those cells, also using next generation sequencing methods adapted for low cell numbers. And by comparing the genetic signature of these cells after therapy with the genetic signature of the cells from the same patient, but at the moment of diagnosis, we might understand or we might get the information on the genes or signaling pathways that are crucial for those cells to resist chemotherapy. If we can identify such genes or signaling pathways, this would mean that we may have really crucial and unique information to overcome chemo resistance to standard and backbone myeloma therapy.
That sounds exciting. Thank you so much.
Thank you.
And it sounds like in your first aim that one of the objectives is, you said typically patients have to wait 2 to 3 years to see if a new therapy is going to work for them, because you enroll people in a clinical trial and you have to wait for the time frame for the trial to be over and then to wait to see if their disease progresses. But what you're saying is if you have the right testing and you can predetermine who will respond and who will not respond to a new therapy potentially, you can cut that time short. Is that correct?
Yes, indeed. This is really one of the objectives of really pushing forward the field of MRD, is to really make possible, and then again, both academia, also pharmaceutical industry, as well as regulatory agencies such as the FDA or the European agency, are really working together to see if there is indeed or not a role for MRD as a tool to really make possible and available new and effective drugs for patients before time. This means that we need to show a clear correlation between depth of response MRD negativity versus MRD positivity and differences in outcome. And so far, this has been really established or confirmed in myeloma, again, by different groups using different techniques. So now I think the time has come to really implement these new and high-sensitive techniques, respectively, in clinical trials to really demonstrate the added value and hopefully in a few years now to come to really, again, help patients, the management of patients with multiple myeloma.
And just a follow-up question, because when you said it had evolved, this flow cytometry test had evolved, and you can go from now 4 proteins, identifying 4 proteins to 8 proteins. What type of proteins are you looking at, and how have these types of proteins been detected before? Were they like RNA sequencing, or is this a totally new type of capability for this assay?
No, many of those. Well, so what we do with the flow is to measure single cells that are in suspension in a cellular fluid, and we analyze the morphology of all those single cells, as well as for the presence or absence of antigens, proteins, in the surface or in the cytoplasm of every single cell. So with this technology, we usually combine proteins, antigens that allow us to identify a cell population of interest, such as plasma cells, if the sample is from a patient with multiple myeloma, and we combine identification markers, proteins, with other markers that allow for the discrimination between a normal versus a myeloma plasma cell, because a myeloma plasma cell has a different genomic profile. A different genomic profile will lead into a different RNA profile, and a different RNA profile will lead into a different protein expression profile, which means that often the myeloma cell expresses antigens in the surface or lacks antigens in the surface that are commonly present in normal plasma cells. And by looking into every single cell and combining the information of the morphology of the cell, plasma cell identification markers, as well as markers that are either absent or present in tumor cells, we are capable of discriminating one tumor cell within now almost 1 million normal cells. And this is the concept of MRD monitoring by flow in myeloma.
And it's taken quite some time to get to that point.
Well, things never come
easy
as we would like. But indeed, in the 90s, at the beginning of the 90s, there was a lot of technological development, I would say particularly from both the Spanish and the UK leads groups. They have really established at the time a 4-color or up to 6-color assay. Then this assay was prospectively incorporated in different clinical trials. At the time also, because this was now 7-8 years ago, clinical trials that did not really incorporate the best drugs that we have available today. Nevertheless, already from those trials, it could be clearly seen that MRD was one, if not the most important prognostic marker. And after seeing those results that were reproduced in different centers and in different clinical trials, this led into this second house of technological development, in which, again, the way we do MRD nowadays is completely different from the way we did it in the past. I can tell you now, and we now have monitored more than 200, 300 myeloma patients only at this institution, and much more in other institutions, with this new assay, And believe me when I tell you that the amount of information that we can now see has no point of comparison, is not comparable to what we have seen before. This means that we are extremely confident and hopeful on a new clinical meaningful of what we are doing in the laboratory.
Okay, excellent. Now, Gary, you had some questions about outcomes between MRD negative, high and low risk patients. Do you want to go ahead and ask those questions?
Yeah, sure, Jenny. Doctor, thank you very much. One thing I was wondering is the 8-color flow cytometry. Is that the same one that's used in Arkansas?
Certainly, in fact, we were involved a couple of years ago, and we are in close collaboration with the Arkansas as well as other institutions in the United States, and they used and they have implemented a quite sensitive 8-color method. The method we are using now is very recent. It is the final product of the last 2 years of research. Therefore, it is not yet fully used in all different laboratories from both Europe and the US as well as other continents. Our goal is really, and of course, being completely reasonable and rational, we know that it is impossible for every single center to adopt the same assay, but it is certainly our goal, after demonstrating the added value of what we have produced in the last 2 years, that a significant number of myeloma centers, top myeloma centers, as well as smaller myeloma hospitals, can really adopt this strategy, which in our opinion is very cost effective. It does not need the sample and diagnosis, which sometimes may be a limiting factor, and again allows for a great sensitivity to measure response assessments. So the Arkansas group, as well as other groups, we now, let's say, periodically meet in workshops and working meetings in which we discuss deeply the way we do flow, and we really make an attempt for many different groups to adopt the same and standardized flow method.
Yes? Yeah, I understand, that you've done some remarkable things with flow cytometry. The Spanish group has just been amazing with regard to that. And is it true that you taught Dr. San Miguel everything he knows?
Quite the opposite. I've been very fortunate to have a boss like Dr. San Miguel, which is not only a boss but a mentor, a friend, and a true friend. And despite all his attempts, unfortunately, in 7 or 8 years that we have been together, I still haven't learned everything that he has to teach me. But I try hard every single day.
Well, he's one of the ones that I consider the great 8 of all myeloma specialists. So I hold him in the very highest of regards. Now, in your presentation, you mentioned that. Or your proposal, you mentioned that MRD negative, high and low risk patients have similar outcomes. Is that did I understand that correctly?
Indeed, and that was a very positive surprise we had recently. Like I mentioned throughout this radio show before. Using the 4-color assay, which was less sensitive, we saw that both us and the UK groups we have seen that the outcome of high-risk patients reaching MRD negativity was in between the outcome of standard risk patients, either MRD negative or MRD positive. It was more or less in the middle of the two standard risk group of patients.
Okay, now here's going to be the clincher for you.
Let me just finish this idea. We now have data from a more recent Spanish trial that started to enroll patients in 2010, in which already there we started to implement 8 colors. We didn't measure yet at that time the same number of millions and millions of cells that we measure nowadays. Let's say that the sensitivity or the assay that was implemented in the 2010 trial was right in the middle between the 4-color flow and the flow we are using today. Nowadays we have a median follow-up for that particular clinical trial that is now in the range of 3 years, so it's still a relatively short follow-up, but very promising. And again, emphasizing on the on the short follow-up, what we are seeing, just by incrementing the number of colors, we are seeing a complete that we are seeing that the outcome of MRD negative patients, irrespectively of the risk by fish cytogenetics, is completely superimposable. This was a very and promising result.
So that's 4-color or 8-color?
Excuse me?
That's with either 4-color or 8-color?
So in 2010, we were already using 8 colors.
Okay. Well, the other, this is kind of the clincher question. Of the high-risk patients that have achieved MRD negative status, is there a treatment plan which has the best outcomes?
I had some hard time to understand your question. Let me see if I understood. If there is a different treatment algorithm according to the MRD status of high-risk patients, correct?
Well, or if you have a high-risk patient and you give them treatment plan A or treatment plan B, and let's say A is RVD transplant maintenance, and treatment plan B is dual transplant plus maintenance. Is one treatment plan provide better results? For example, I know that you guys did something. I think it was the Spanish group did something by, I think it was RVD, and then you had CT. You alternated old drugs, one right after the other, and came up with some pretty great results. I'm not sure if it was your group or it was another group, but I saw it at ASH, and I was wondering if there's a plan or some kind of a treatment plan. Nobody's come up with the plan, that resolves high-risk disease. So is there anything that gives a better response that you've seen, given your experience?
Well, that's a very, very big question, perhaps one of the most important. Let me tell you that the study that you are describing, the alternating study that incorporated all or the majority of the effective and available agents for the treatment of elderly myeloma patients, was indeed the study that I was just describing, that we used the 8-color method and we were seeing superimposable results for high-risk and standard-risk patients reaching MRD negativity. So it was that specific clinical trial. Now, regarding your question, which again is a critical question, I'm deeply sorry, and being completely honest, unfortunately there is no answer. And the reason why there is no answer is because we need to be extremely cautious on the way we move from a laboratory biomarker into a diagnostic tool or a treatment decision tool. A treatment decision tool is equally important as a new drug. Equally important. This means that now that we have further increased the sensitivity of our methods, now that we are standardizing the methods, now it's really the time to implement such methods in all possible clinical trials, to do the biology, to address the biology questions regarding MRD, in order to collect prospective data from clinical trials among patients receiving different treatment schemas in order to address your question. Before we collect that data, it would be, I would say, very incautious to address the question, because it's really addressing a question without having consistent data. So first,
I guess my point would be is, in that trial, as I recall, there was really one of the things that was interesting is in that trial where you sequenced these drugs, the difference between the high risk and low risk group was non-existent.
Yes.
As far as outcomes, which is remarkable.
Indeed. And and again, emphasizing that the follow-up of the study is still in its midterm. So definitely more mature data will be needed to really make a final statement on that particular analysis. But yes, this trial, as well as I would say other clinical trials, are really showing that the outcome between high risk and standard risk patients, the poor outcome of high risk patients is not yet fully overcome, but is being ameliorated. This means that, and this is my personal point of view, that as therapy becomes more and more effective, the way we call high risk now may be different from the way we will call high risk in the future. Plus, this will also mean that perhaps, and everything is complementary, but perhaps more important that risk stratification at diagnosis, because therapy has become so much effective, perhaps more important than that would be really a sensitive monitoring of response to therapy.
Okay.
So and really to make a final argument on your question, and again, really sending the message that as we move along very slowly, as it needs to be done, we cannot rush things into precision medicine. We need to make very safe questions. So, for example, this research program that was more or less described in the letter of intention would take place, for example, in a clinical trial, for example, that is being conducted by a Spanish group, in which, for the first time ever, we make a very precise clinical questions regarding MRD, which is not at the beginning of the treatment, but at the very end of the treatment, after a period of maintenance therapy, we make the question of whether or not maintenance therapy can be stopped according to the patient's MRD status. So patients that are MRD negative will stop maintenance, whereas patients that continue to be MRD positive, they will continue to receive maintenance therapy. This will allow us to understand if sensitive MRD assessment not performed one single time, but performed throughout the patient's treatment, after induction, after transplant, after consolidation, and then after maintenance, and using sensitive techniques can perhaps tailor the duration of maintenance. So again, really emphasizing on the prospective nature of the clinical questions, as well as to be very cautious on the clinical questions we postulate using a laboratory biomarker.
Okay. Well, Doctor, thank you so much. One other thing is that you said that if you're not MRD negative, there's clones left over, and you're going to take a look at those clones, right? Whether it's in high or low risk.
Yes.
And see, and evaluate those clones to see what the difference in those clones are. And this is actually a question from I think the Dallas Myeloma Group, the multiple myeloma support group there, who asked me to ask this question, and that is how will this evaluation lead to new treatments?
Okay, so that's a very pertinent question, a very good question. By understanding the genomic signature of those cells that resist after therapy, and then again the importance of really studying the patients longitudinally, because if we only studied that cell alone, the information would be limited. We need to compare that MRD cell with the baseline cells before the patient has entered therapy, to really understand the genes that were deregulated, as well as the kinetics of clonal selection by looking into the mutations. Now, if we understand, and there is consistent data from patient to patient suggesting that for a particular treatment schema, there is always, or in a significant number of patients, deregulation, downregulation, or upregulation of a given gene or a given pathway. This means that we can then move, for example, into in vitro systems using myeloma cell lines to manipulate those genes that we identified in the precious patient samples, develop in vitro myeloma cells with the signature of MRD cells, and then test in vitro how to overcome the chemo-resistant profile of those MRD cells, either by testing new drugs, targeting new genes that were so far not targeted in myeloma, or by combining the many different and available agents.
Thank you, Doctor. Jenny, I think there's someone else ready to ask some questions. Is that correct,
yeah.
Jack?
Jack, you had a couple questions.
I've got one more, but only if I have time.
Jack, would you like to go ahead with your questions?
Yes, I'm on the line. I really appreciate this conversation, although it's quite difficult, and I apologize if I'm asking similar questions to what I've already been asked. I want to understand with respect to the 8 antigen or color markers that you're using for flow, are they the same? Have we standardized those 8 now? Are they the same that go with the Black Swan initiative that different institutions now are being trained to use those same 8 markers?
Indeed. The platform or the assay we are currently doing today is the assay that we helped developing within the Black Swan Research Initiative.
Great. And because I know at one point they talked about 10 markers and things like that. So, and I'm sure you've already done some interesting research in your labs where you're looking at effectively the degree of resistance in the high-risk subgroup. and you're looking to find whether or not there are some potential targets that make these cells more resistant. I'm wondering if you've already found any, or are you looking at any targets, and might we already have some drugs that have been developed to tackle these targets in other cancers?
We have now, we have started this concept of MRD as a reservoir of chemo-resistant cells, and therefore a clone for intensive research. A couple of months, let's say 1 or 2 years ago, and in fact, and this has been presented in the American Society of Hematology meeting, I would say 2 years ago, last year also, in which, and at the time, again, limited by less high throughput techniques, we were able to characterize for the first time ever the MRD cell. And we have detected that the phenotype is slightly different, as well as the genotype and the transcriptome. But then again, this using, I would say, intermediate sensitive platforms. And we are now also trying to validate this model as, again, the model for identification of new drugs or new drug combinations. But what we want to do now is really to synergize state-of-the-art technology to address this question. So only now, with very sensitive flow, we will be able to sort those cells with high purity, and also only now with the DNA and RNA sequencing tools adapted for, I would say, almost single cells, we will appreciate all the genome and the transcriptome of those cells. So hopefully this will give us information that so far has never been collected. We believe that in parallel to all the efforts that are being done so far to sequence the myeloma cells of patients with newly diagnosed myeloma, we believe that equally important will be to perform those sequencing efforts in clones that are typically not characterized, which is the circulating tumor cell as well as the minimal residual disease cell. We also believe that, and this to some extent might even be rational, for example, and now getting back into the newly diagnosed setting, it is of common knowledge that, for example, gene expression profile signatures are of great value to discriminate between standard versus high-risk patients, but they are different from scientific group into scientific group. And our personal feeling or belief in this regard is that those signatures are different because they were obtained with different treatment schemas. They work in different trials, but they are obtained with different treatment schemas, which means that most likely this strategy of trying to overcome the chemo resistance of MRD cells will be specific to the common backbone drugs being used for each particular clinical trial, because certainly the nature of ultra chemo resistant cells will slightly differ according to the different drugs being used.
Thank you. I have one other question, and it kind of requires you to think about a graph Along the x-axis, you have standard risk and high risk. Along the y-axis, you have MRD negative, and above that, MRD positive. And I think the genomic significance that you're looking at in your letter of intent focuses on the top part of that graph. In other words, the MRD positive for standard risk versus high risk patients. And I wonder if ultimately one will need to look at more. For example, are we sure it's better to be a high-risk MRD negative patient than it is to be a standard risk MRD positive patient?
Those are, I believe, Jack, are two different questions. So regarding the first, and again, looking into the outcomes with a relatively short follow-up, but of more recent clinical trials, it seems that the outcome of patients with high-risk disease reaching MRD negativity is indeed superior as compared to the outcome of patients with standard risk disease and not reaching MRD negativity, in other words, remaining MRD positive. So that would be the answer for your last question. Regarding the first question, absolutely. Equally important as to characterize the biology, the genetic landscape of ultra-chemo-resistant cells from high-risk patients will be also to do the same approach in standard-risk patients. Also to understand the difference in the kinetics of chemo-resistant between the two patient cohorts. So this will be crucial. Also, and this should also be a research priority to all myeloma scientific groups, as you are probably aware, depth of response is a key prognostic factor, but it does not afford the same answer to all the patients. In other words, there are patients that remain MRD positive and nevertheless experience very long-term outcomes, which means that we need to understand, again, the genetic profile of that resistant cell in order to identify clones that are left there after therapy, but that will behave like MGUS clones. And these can be there for many years. The patient may even have persistent MRD, may even show a positive M spike in the serum, but the patient will be progression-free for many and many years. And really, to dissect or to discriminate between an MRD positive patient that will unfortunately progress sooner from those that fortunately progress much later on in time, if they even progress at all, we believe will arise from two different research fields. Again, first, and this is what we are discussing here, to study the biology of those cells. Secondly, to perform in-depth research on the immune system, to understand how the patient's immune system are able or not to control residual disease levels after therapy.
Thank you very much, Dr. Paiva. I wish you the very best of success for all myeloma patients, and I'll put myself back on mute, Jenny.
Okay.
Thank you.
Okay, great. Thank you, Jack. Pat, did you have any questions before I have a couple things I would like to ask? Okay. Dr. Paiva, I wonder if you could outline
Jenny?
for us. Go ahead, Pat.
Oh, sorry. See, I'm not familiar with the mute button. So I thought it was off. It was on. Sorry.
Okay.
Good. Now I was going, can you hear me? Can you hear me? I just have one question, and it's related, but it's not. I mean, I think in a basic myeloma patient's mind, it's probably related, but they are two different things. Is any of the work you're doing, doctor, helping to helping doctors monitor non-secretors so that we can more closely follow the progress of their myeloma?
Absolutely, and again, I'm really appreciating this now already 1 hour of debate, scientific
goes by
debate.
quite fast.
Because this has been tremendously stimulating for hopefully for all of us that are online. That's a very good question, and in fact, you are probably aware that just recently, in 2014, the diagnostic criteria for myeloma was updated by the International Myeloma Working Group, and for example, one of the updates was to really remove the need of an M protein to diagnose a patient with multiple myeloma, realizing the existence of the non-secretory patients. Now, for the non-secretory patients, there was always, from the last 5, 6, 10 years, the possibility, as you are probably aware, of using the free light chains.
Sure.
They are more sensitive as compared to standard electrophoresis or immune fixation methods to detect the M components. So the free light chains afford information on how to measure the M protein, let's say in around 50% of the non-secretory patients, but certainly to measure the clonal cell in the bone marrow affords information in virtually all the patients. It should be emphasized that from a phenotypic point of view, from a cellular point of view, so far we have not seen differences between the phenotype, that is the way we see the cells, between secretory versus non-secretory myeloma patients, which means that the technique or the method we developed, the method we use, can be commonly applied, equally applied to both secretory versus non-secretory myeloma patients.
Oh, thank you. That helps. And Jenny, I want to commend you. I know a lot about multiple myeloma, but I'll tell you, this is pretty complicated stuff, and you do a great job helping to simplify things. And the panel's done a wonderful job, I think, helping patients understand what's going on, because this is pretty technical stuff.
Yes, it is, and we appreciate all the good questions. They're excellent, and your great responses, Dr. Paiva. So what I hear you saying overall is that you want to both capture the baseline through some standard genetic tests, and then you want to use this flow cytometry, the next generation, to compare it against that baseline to see what genes are regulated, how does myeloma change and morph, so you can create a plan of attack to manipulate the genes that are causing those problems. And I think one thing that you said that really stood out to me is that you want to test a higher amount of cells so you can look at single cells. And I would say to patients who this is a little complicated, but to patients that need to understand this better, that is so important, because that single cell can become the problem, over time. It might not be a problem when you first get hit with your induction therapy or even your transplant, but after that's finished, if you have that cell left over, that new cell that maybe wasn't very aggressive before can become the single problem. So in my mind, that's why this work is so important.
Well, thank you. For us, it has become, I would say, a very passionate research area of our group. Again, we are really committed to study two very rare cells. The first is the circulating tumor cell, which is not contemplated in this research program, but we are deeply committed to study that cell, because we believe that the peripheral blood circulating tumor cell is the one responsible for the disease dissemination. Secondly, to the MRD cell, And if for a second now we close our eyes and we try to not think on the prognostic significance, on the potential role for treatment decisions, tailoring therapy, and we only think on biology, if we close our eyes, we can imagine that, or we can envision that these cells are very few, only a few have the true potential to resist such effective treatment strategies. These cells are likely quiescent after therapy. They are not producing, in the majority of cases, the M protein, the monoclonal protein. These cells can be dormant for many years, but then again, these cells are the ones responsible for the patient's relapse. These three descriptions, or these three features that I just mentioned, have been, let's say, the three hallmarks of the worst cancer cell that has ever been postulated, which are the cancer stem cells. So we truly believe that this MRD cell has all the features of the cancer stem cells, and if we are either able to really reduce their levels through treatment, if we are able to monitor them very sensitively, and now if we are able to study or to unravel the biology of these cells, hopefully this could be of great value to this disease as well as to other diseases in hematology. So we are truly committed to this research program in our laboratory.
Okay, perfect. My final question is just about, you've talked about your research aims, what you're hoping to accomplish. Could you outline for us the specific milestones in this research proposal, and then just give us an idea of a general amount of budget that you would need to accomplish those specific milestones?
The milestones, give me 5 seconds to give like 3 milestones for the this research program. So the first would really be to establish ultrasensitive MRD negativity as a surrogate for long-term disease control in high-risk myeloma, as well as a biomarker for early drug approval. This will be the first. The second will be to define for the first time ever the genetic signature of MRD cells in both high risk as well as standard risk patients. The third milestone would be to establish the MRD cell as a new model for identification of novel targets as well as drug development. I think this would be the key 3 milestones of the research program.
Mm-hmm. And then do you have an idea of what you would need budget-wise to accomplish those 3 milestones?
Again, being very, very reasonable and trying to be cost-effective in any way we can. And in fact, the way we developed the flow assay was to one of the person that is online asked about the number of colors. I don't remember now his name, unfortunately, and I'm sorry for that. But he asked for the number of colors, 8 or 10 or more. Certainly, the more the better, like always. But we need to commit, and we need it to be cost effective, so it can be available for all the patients, and we need these techniques to be available for all the centers, those that are more specialized, perhaps with also more resources, as well as other centers that don't have the resources of big groups. This means that all the biomarkers that we try to develop here are really with that purpose of being tremendously cost effective. So if we want to study the phenotypic characteristics of the diagnostic cell, of the myeloma cell at diagnosis, as well as the MRD cell that is there after therapy, and if our goal is to perform this longitudinal comparison, this would be 2 time points in a series of 100 patients, for example, comparing 50 high-risk patients versus 50 standard-risk patients, which means again that a cytogenetic characterization must also be performed at baseline to identify the 2 patient cohorts. Then RNA and DNA sequencing studies to be performed both at the baseline as well as the MRD level, and then also for in vitro studies to develop the model of MRD as a platform to develop new drugs or new drug combinations. I would say that this would be more or less in the range of $1,500 to $2,000 per patient.
Okay. And you're trying to study 100 patients?
Yes.
Okay, perfect. Well, that gives us an idea of scope, I guess. So it's very helpful to have that. Well, I think that one of the most exciting things about your project is that for the first time, this flow test is weighing in on the biology of the disease, which has never really happened before. And the biology of myeloma is so complex, and even within a single myeloma patient, they can have several different kinds of myeloma cells within their own body. So that the more testing and the more detailed and the more biology it's studying, I think we are far better off having that.
Can you rephrase the question?
Oh, no, I was just saying how significant I think it is that for the first time you are studying the flow cytometry test is studying the biology.
Hopefully, again, we are extremely hopeful that this new approach may really afford information that was never there before.
Right.
And then again, I think that really the laboratory as well as the biology should keep with the same pace as the improvement in treatment options. And it is our opinion that again, and perhaps coming back from where we started with this radio show, I think the time has come to really improve response criteria and the biology regarding response criteria in the same level as differential diagnosis, risk stratification, and treatment options have been improved in the last 5 to 10 years.
Absolutely. Well, we agree, and we're so thankful for the work you're doing, the study that's been done by you and your group. We're just very grateful because it's helping discover many new approaches and targets for myeloma. So, Dr. Paiva, we've kept you way over time today, but we think it's worth it. So we're so thankful for your participation today. Thank you for all you're doing.
No, it was my great pleasure. This has been one of the most exciting and stimulating hours of my research life, and again, I can assure you that all of us here in the institution, in our institution, work hard every single day to really improve the lives of myeloma as well as the patients with other hematological malignancies. It is our really great pleasure to participate in these events.
Well, thank you so much. We're very fortunate that there are people like you working so hard for us. So thank you. Now, we'd like to thank you for listening to Myeloma Crowd Radio and the new MCRI series. We know that patients can help support the discovery of a cure, and we encourage you to become more involved.
Step into the world of power, loyalty, and luck.
I'm gonna make him an offer he can't refuse.
With family, cannolis, and spins mean everything.
Now, you wanna get mixed up in the family business.
Introducing The Godfather at ChumbaCasino.com. Test your luck in the shadowy world of The Godfather slot.
Someday, I will call upon you to do a service for me.
Play The Godfather, now at ChumbaCasino.com.
Welcome to the family.
VGW Group. No purchase necessary. Void where prohibited by law. See terms and conditions. 18+.